Good evening, everyone. So today's session, we are concentrating on how to perform or how to do QSAR. That could be a 2D QSAR, a 3D QSAR, or 1D QSAR. Now you will get to know much more about what is this 1D, 2D, and 3D. And today we are using open software that is freely available, which is called PaDEL. PaDEL is from a group, and PaDEL is a software mainly to generate descriptors, right? Fingerprint descriptors, 1D, 2D, and 3D descriptors. They do not generate any QSAR models. Please keep that in mind. It is just a software to predict the properties or descriptors. It will also help you to convert your 2D structures to 3D structures. If you want to retain your 3D structure, that is also possible. For QSAR modeling, we are using software called as QSARINS. QSARINS from Professor Paula Grammatica. I would like to acknowledge her now. Thank you so much for her giving me the permission to use her licenses. The software is completely free for academics, but you cannot simply get the access to the software. You should be currently working on QSAR, and you should have published many papers. And if you give her some references which you have already worked on with your work, yes, she's willing to give you access to the software. So it is not like simply use it for free. You need to prove that you're actually working on this particular area seriously. I've been using QSARINS since 2012, published a few many papers. Recently also published a paper with my student Ambli. So today Ambli will be taking over the hands-on session for PaDEL and QSARINS. One more thing which I want to specify today is that we will be using Excel a lot for plotting, for validation, for applicability domain. Hope you remember, whoever have attended my session on do's and don'ts of QSAR, which is there in my YouTube channel. There I have explained many, what to say, precautions actually, right? So those things are will be discussed here. If anybody want to know my YouTube channel, this is the YouTube channel. If you go there, there are plenty of many more other videos. So if you go here, it's here, what is QSAR, do's and don'ts. Okay, so that will be accessible. And there are many other videos also, like 9 extensions for Cheminformatics, network polypharmacology, then some e-conferences on COVID, as well as something on overview on like ADME, then molecular docking using Autodoc, then yesterday's session on molecular docking, dynamics using GROMACS and the Jupyter Notebook. I heard that many of them found it easy so that they can easily run the GROMACS MD simulations, and a detailed tutorial on modular software for doing homology modeling. So this is just to give you a glimpse about the channel, where if you want to have a little bit understanding. So I would recommend you to please go through the theory part of QSAR first, and then only go thorough into your research using PaDEL and QSARINS. So as usual, I wanted to say that kindly switch off your webcam for better buffering. You should be in mute mode, unmute to engage in discussions, but kindly tell your name before engaging in discussions via audio. All your questions could be shared by a mentee. The advantage is any question shared by a mentee is anonymous. So even if you're curious and to ask questions, feel free to shoot any questions. And please answer a few questions in the next slides. And you can go to Menti and give this code 162066 to answer the questions for the next slides, okay? So this is just to understand what is your area. I can see a few previous entries also, but please go ahead to just understand what is your area of or field of research or studies, so that to just have a better brief on where we to emphasize more. So I could see more of bioinformatics people, and also molecular modeling, and a small amount of, yeah, around 20, close to 20% of pharmaceutical sciences like pharmacology, medicinal chemistry, pharmaceutical chemistry, and a small number of biotechnology also. Great. I think, yeah, okay. So equal number is coming up. Uh, I can wait for 2 more seconds. No, I don't want to waste too much time here. So if you can make it fast, it is good. Otherwise, we'll move ahead. I think we are fair enough. We are having a better understanding. Okay, moving forward. So going here, I wanted to understand how many of you have really used QSARINS, just to make an understanding, so that it will be useful. How many of them? Okay, I can see one person is already using it. Okay, the other person has not heard about it. Okay, anyone else? Okay, so there are more people who have not heard about it. Okay. So I think even if you're working on QSR, you might be using some paid versions or some other statistical methods, or probably maybe. Yeah, but QSR, as I told you, it's a good piece of software, but in order to get access, you really need to showcase that you're seriously working on that. Great. There are two people working on QSRNs, but I can see more people not working and not using QSRNs. Great. Now this will be hopefully useful. Now, okay, I can see that more are not using QSRNs. Great. Thank you. So going for the next one, this is just to understand what is your confidence level on a certain area that you're looking at. For example, QSAR, a detailed understanding about its theory. So this you have to rate it from 1 to 5. You can rate all of them. It's not that only one. You can rate all of them. At the bottom of your page, there's a submit button. Okay. So here I'm just trying to understand how detailed you know the QSAR theory. That is very, very important. There are several reviews are there from ACS, and recently Paula Grammatica has written a review, a very wonderful review. I can share those links in the YouTube description once this video is done. I also understand many of the community is using Open 3D QSAR. Of course, it has its own learning curve, but if you are there, it is very useful. And for modeling purpose, like generating the statistical model or SVM or machine learning models, many of you might be using Veka, R program, SPSS, statistic, or any other statistical programs. Then again, QSARINS or Codesa Pro. I was working with the group, Professor Alan Katruski and Matti Karlsson, who are the authors behind Codesa Pro. So yeah, and also Excel data management. It's very important. You need to know a bit detailed about Excel data management, including charting, how to create charts and all others. Even in Excel, you can do a bit of statistics, but not multilinear regression, but normal linear regression can be easily done in Excel. By default, it is not enabled. You have to go to plugins and enable the statistics mode, and it works fine. Anyway, so already 6 people have given in the earlier slide. There was around 9, but yeah, here only 6. But anyway, it's a good. So I can see the audience still having a good understanding about QSAR. So only help you need is probably more of how to work on QSAR with combination with PaDEL. So let me tell you, in QSARINS itself, the latest version, they have PaDEL inbuilt in it. So you don't require a PaDEL separately to generate the descriptors. So you have PaDEL inbuilt within the QSAR. But today, Ambali, who is my student, will be more focusing on PaDEL separately and QSARINS separately, so that you can use a descriptor from PaDEL for any other software for modeling purpose. So as an overview, today we'll be discussing about the softwares which I mentioned earlier. We all should refer to only open source references, and no promotions of paid products are entertained. And we are not going to compare any paid products also. Of course, you can ask questions, but We will be talking more about concentrating on this software which we are talking about, but not doing any comparison. All the opinions and discussions are personal views with no connection or relation to current employees or corporates of any other individuals. So we are going towards the demonstration. So the installation, then PaDEL and QSARINS and the result analysis. So from now, I'm moving to Ambli, and she, as I told you, we recently published a paper on life sciences on anti-melanogenic agents. There we used a combination of QSAR, molecular docking, then pharmacokinetics, and designing new molecules and predicting its activity. We and a nominal MD, but we didn't go for synthesis and testing uh, because of funding issues and resources. So we didn't go for that, but we made sure with medicinal chemists those newly designed compounds are 100% synthesizable, and we can always share the synthetic pathway, the protocol, if someone is interested to take it move forward. So we do have the pathways for those new molecules. We can always share it with anybody. Anyway, so that's where today we will be going up. And while the session is, I can see already 4 questions are there. So this will be during the Q&A session, we will take up the questions in menti.com. When you give the code 162066 or the links that I've shared, you can ask a question at any time. All the questions will be taken care of during the Q&A session. So now I am sharing it to Ambli, and she'll be taking the session. So let me tell you, being the sessions a bit longer from the previous experience, the speaker getting so much tired. So we made few recorded videos, but we are here. Ambli is also here. I myself will be here. So in between, if you have any questions, please feel free. So this is to just make the things much productive. Okay, so let me share the screen so that she will be able to do that. Just a minute. Okay, so I hope you're able to see the screen. And let me just check.
Good evening, one and all. Today we'll be having a software demonstration on PaDEL and QSARINS.
There's some
Before
sound
going
issue.
into the details regarding the software, I would like to give a brief introduction on QSAR. So QSAR is quantitative structure activity relationship. This employs mathematical modeling approach to predict the activity of chemical compounds. So the activity could be biological, physical, or chemical. So the entire concept of QSAR can be explained by using simple equations. That is, activity is
Uh,
the function of physical chemical properties plus or minus K. So here, if we discuss about this equation, we could see that there are two important attributes. The first one is the biological activity, and second one is the physical chemical properties. So, what is the physical chemical properties? These are nothing but the structural features of chemical compounds. So, structural features in the sense it could be the presence of rings, number of atoms, number of bonds, connectivity between atoms and all. So, by utilizing a QSAR model or by building a QSAR model, we will be identifying the impact or how the structural properties of chemical compounds can contribute to the biological activity, whether it can enhance the activity or whether it can reduce or produce a deteriorating activity, deteriorating effect for the activity. So that is all about the structural features. As I have mentioned earlier, the QSAR modeling is based on a mathematical modeling approach. So all the chemical information or the structural features encoded within the molecule will be converted into numerical values. These numerical values are depicted as molecular descriptors. These descriptors represent the independent variables in QSAR modeling. So the next important attribute is the biological activity. So how we will get the biological activity? How it is being expressed? So for example, if I have 10 novel compounds and I want to check whether my compounds are potent for acting as antioxidant agents. So I will have to perform certain bioassays to confirm the potency of these molecules to function as antioxidant agents. So bioassays for antioxidant activity could be TPPH assay, ABTS, or FLAP assay. So once this bioassay is performed, the potency of the molecules will be expressed as IC50 values. What are IC50 values? IC50 values are nothing but the concentration of a drug required to produce 50% of inhibitory activity for a particular biological process. This is half maximal inhibitory concentration and it is a measure of potency. So after performing bioassays, we will be getting the IC50 values and these values represent the response variables or dependent variables. So once we are done with the molecular descriptors and once we are getting the response variables, we can go for the QSAR model building. We have different methodologies for QSAR model building. So here I have listed some of them. Genetic algorithms, multilinear regression, principal component analysis, partial least squares, neural network, random forest, support vector machine, and Gaussian process. So out of all these methods, MLR is one of the earliest methods for constructing QSAR model, but it is still one of the most commonly used one. MLR, which is also known as multiple regression, it is a statistical technique that helps us to identify the impact of dependent variables on dependent variables. It helps us to identify or it helps us to come up with the linear relationship between the independent variables and dependent variables. So in QSAR, independent variables are molecular descriptors and dependent variables are the response variables. So here if we consider GA, that is genetic algorithm is employed to identify the most significant descriptors which could correlate with the biological activity. So in our evolutionary biology classes, we might have studied about the survival of fittest. That is a concept in evolutionary biology. That means that the populations which are well adapted to a particular environment will survive better than the others. So similarly, in this algorithm also, we are trying to identify the most significant descriptors which could correlate with the biological activity of chemical compounds. So there are different other algorithms are also there. So today we'll be mainly discussing about the MLR based as well as GA based algorithms including QSAR model building. So this is the overview regarding the QSAR. So now in this slide I'll be explaining the QSAR methodology. So the very first step for building a QSAR model is the dataset construction. So for a dataset construction, we will need different sources. We will have to identify our ligands or chemical compounds from different sources. It could be from literature, it could be from the databases and all. So before going into the details, for QSAR model building, minimum of 40 compounds are required. So if you have more number of compounds, you will be able to generate predictable and reliable QSAR models. So in general cases, we won't get a minimum of or 40 compounds from a single paper. We'll have to identify different papers which in which they have reported the IC50 values as well as the structure. So how will you do that or what are the conditions for that? For example, if I'm doing a QSAR model for anti-tyrosinase activity, so I have searched literature and in one paper I've got around 25 compounds with their IC50 values. And there is another paper which they have reported 30 to 40 compounds which is having anti-tyrosinase activity with their IC50 values. So in this case, we can include the 25 compounds from the first paper and the 40 compounds from the second paper, provided that they are characterized using similar bioassay. Okay, so if there is any difference in the bioassay, we won't be able to merge this two datasets or the compounds from two different papers. So they should have similar bioassay. And the second criteria is similarity in their units. So usually the IC50 values will be expressed in micromolar or nanomolar units. So if we are merging the compounds from two different papers, we will have to check for the similarity in their units. Okay, so after getting all this criteria done, the next step is to draw the chemical structures. We have different platforms to draw chemical structures. One of them is Marvin Sketch, which is provided by Chemaxon. So in Marvin Sketch, you will be able to draw the structure, you will be able to generate the 2D coordinates, 3D coordinates and all. I'll be explaining like how to draw the structure and all. So once your chemical structure is ready, then the next step is the geometry optimization. So what is the rationale behind doing a geometry optimization? So geometry optimization is performed to make optimum energy of a molecule to minimum. Okay, so we know that energy of a molecule is correlated with their stability. So we need to get a more stable conformation of the molecule. So to like more detailed explanation is that for energy minimization, like why we are doing geometry optimization is that we'll be identifying the minimum energy conformation of a molecule, that is the atomic force on each atom that is acting on each atom will be acceptably close to zero. Okay, this is related the stability of chemical compounds. Then after the geometry optimization, the step involved descriptor generation. So as I have mentioned in the previous slide, descriptors account for the chemical information encoded within the molecule, which is converted to the numerical value. So we have different platforms, online tools are available for descriptor calculation. One of them is PaDEL, other one is Teresa. So I'll be showing how to prepare files for each of these softwares and how to run the calculation. So once this descriptor generation or descriptor calculation is done, we have to go for feature selection. So once the descriptor calculation is completed, you can see there will be a pool of descriptors or large number of descriptors will be there. But out of this large number of descriptors, we will have to find out the most significant descriptor which could be correlated with our biological activity. So this is being done by utilizing several algorithms. GA genetic algorithm is one of the methodology employed for feature selection. Okay. Then after that, QSAR modeling, and this is followed by the validation. So validation, there will be two types, that is internal as well as external validation. Internal validation will be done based on by utilizing the molecules which were used for QSAR model building, but external validation will do with a totally new external set of chemical compounds. So this is the entire methodology for QSAR. So now we will see what are the sources of chemical structure. So as I have mentioned, the literature is one of the most important source where you can find the chemical structures, the bioassays, and the activity values. Okay, so once you are doing or once you are done with the literature survey, you got adequate number of chemical compounds, but you are interested to study the molecular features of the chemical compounds, or you are interested to study the pharmacological features of chemical compounds, you can navigate to the databases such as PubChem as well as ChemDel. So I will show you how we can get the data from PubChem and ChemDel. So this is the platform of PubChem. So here if we check we can explore different chemical compounds. We can have the query compound here. So for example, I'll show you, I'm typing gallic acid. Okay, this is a natural compound which is predominantly found in many plant-based formulations. So if you click on gallic acid, you will get the compound report. The entire profile of this chemical compound can be identified from the PubChem. So if you go to the right hand side, you can see what all information you can extract from this particular page. Okay, you can find out the structures like the 2D, 3D conformations you can identify, and you can get the molecular formula of chemical compound, the synonyms, molecular weight, and the 2D structure is available. So if you're going to if you have identified certain chemical compound from the literature, and if you identify certain ambiguities in your structure, you can always cross check the structure from these databases. Okay, so you will have the 2D depiction of your chemical compound here, 3D is there. There is an option to download. Okay, downloading SDF format, you can download and keep. Similarly for the 3D conformer. And also we have like different, the IUPAC name is given, the inch key is given, canonical smiles is given. So all the relevant information related to a particular compound, you can get it from the PubChem database. As well as we can have the details about the chemical vendors. If you're going to purchase this chemical compound and if you're going to do a particular bioassay, you can always check this data about the chemical vendors, the drug medication, the pharmacology profiles, what are the patents associated with it. Every details you can easily get it from the PubChem database. Okay. Similarly, the other one is Kendall. So in Kendall also, you will be able to search the particular chemical compound. So here one of the example given is imatinib. So the commercial name of imatinib is Glivec. So Glivec will help us to, Glivec is a chemical compound which was approved by uh, the FDA, okay, approved by FDA, and it is utilized for the treatment of blood cancer, okay, CML, that is blood cancer. So now, if you check the records, so here different IDs are available. So if you click on any one of this ID, will get the compound report card. So similarly, similar to the PubChem database, if we check, we'll get all the relevant information related to a chemical compound. What is the synonyms associated with that? What is the molecular weight, molecular formula? Then we can also identify the molecular features. Okay, so molecular features like we can identify the drug type. Here it is a synthetic small molecule, and this is related to Lipinski's rule of five. So when we are considering the lead compounds, all the lead compounds are like it is not mandatory to have the Lipinski's rule of five for the lead compounds. Okay, so the Lipinski's rule of five is mandatory for orally bioavailable chemical compounds. So if we see the case, this compound, this Gleevec, is an orally bioavailable drug, and so it should pass the Lipinski's rule of five. So it will give you information about the routes of administration. It will give you the information about the chirality of the molecule, like whether the molecule is superimposable or not, whether the molecule is having the chiral centers or the asymmetric carbon atom, and all it will give. And the drug mechanism. Here, if we go, this molecule is acting as an inhibitor for three targets like stem cell growth factor receptor, tyrosinase protein kinase ABL, and platelet derived growth factor receptor beta. And there are so many drug indications for these all diseases. Okay, so the references is taken from the clinical trials. Okay, so the similar compounds also you can get. So the compounds with the 85 percentage similarity is shown, and the metabolic profile of this chemical compound is also shown here. Metabolic profile in the sense how this chemical compound is metabolized by the cytochrome P450 isoforms, or what all isoforms are metabolizing this particular chemical compound. And here if we go to the bioactivity summary, okay, so here you can see the IC50. So if you click on this particular window, what we will get is that the IC50 values of this chemical compound. Okay, so here you can see the IC50, the standard value of IC50, and we can also get the assay, like what kind of assay is utilized for the characterization. We can also get the
Excuse me,
details
Amli.
regarding.
Just a minute. It seems the clarity is not so good, so let me just check it out. Um, yeah, they're all fine. Okay, so let me share it with you. Go ahead. Just a minute.
This
Okay.
essay. So if you go to Chembil and if you search a particular chemical compound, you will get the entire details, okay, regarding their biological activity, their molecular targets, and their what is the clinical trials being done or not, all the chemical features like IUPAC name, the smiles, everything you will get. So if you find any ambiguity from the for the compounds that is published in literature, you can always cross-check it with some of the databases like PubChem and Chembil so that you will get a well-curated data for generating your QSAR model. Okay, so this is all about the sources of chemical structures. Okay, then moving on, how to draw the chemical structures. So we have a software platform that is called Marvin Sketch. It is provided by Chemaxin. So in Marvin Sketch, you will be able to draw the chemical structures. Okay, you will be able to convert the 2D coordinates to 3D coordinates. And if you are not well versed with drawing the chemical structures, but if you have the IUPAC name, you can generate your structure by giving the IUPAC name also. So during the demo session, I'll be explaining how to draw, how to convert the chemical structures, everything. So we have the platform Marvin Sketch for drawing the chemical structures. We have some other platforms also like ChemDraw is also there for drawing chemical structures. So descriptors, much more detailed information about descriptors. So as I have told, it is the chemical information encoded in a molecule. So we have different classes of descriptors are there. One-dimensional descriptors generally represents the atoms in a chemical compounds. While 2D descriptors helps us to understand the connectivity of atoms in a compound. Okay, and 3D descriptors are there. So this 3D descriptors will help us to understand or aids in the understanding of conformations of our compound. Okay, the steric factors, the shape, everything will come under the category of 3D descriptors. Okay, so here I have given a pictorial representation. So we have different classes of descriptors are there: constitutional descriptors, topological descriptors, geometric descriptors, as well as the hybrid descriptors. Okay, so constitutional descriptors generally represents the atom count as well as the bond count in a particular chemical compound. While topological descriptors represents the chain, like the chain of the chemical compound, the number of rings and all. Geometric descriptors needs 3D conformation or it will need the 3D structure of chemical compound. So the calculation of geometric descriptors will take some time compared to the 2D descriptors, and we will have hybrid descriptors like polar surface area, electrotopological indices, shape descriptors and all. Okay, so these are the different classes of descriptors. Okay, so in this demo, I'll be showing the descriptor calculation by using Codesa Pro as well as PaDEL software. Okay, so first of all, I would like to explain about the Codesa Pro software, and during the demo session, I'll explain how to prepare files for the for calculating or for running the Codesa calculations, and then we'll move on to the QSAR model generation. Okay, so Codesa Pro, I'll show you how to navigate to that particular software. So this is the homepage of Codesa Pro software. So this software The authors of this software are Alan Roy Katchowski, Matty Carlson, and Ruslan Petrucci. Okay, so we can see what are the classes of descriptors which are being calculated by this software. Okay, constitutional descriptors are there, topological descriptors, geometrical, electrostatic descriptors, polar surface area related descriptors. Then we have MO related, that is more related to chemical bonding, molecular orbital theory related descriptors, quantum chemical descriptors, and thermodynamic descriptors. So if we want to see what is the each of these descriptors, or what is the physical meaning of this particular descriptors, you can go to this page and you can see the physical meaning of this descriptor. So each of the descriptors has got its physical meaning, so we can easily understand this from the home page, and the manual is also given there, so you can have a look onto the manual as well. Okay, so this is the Codesa Pro software. So So this is the class of descriptors for the Codesa Pro software. Okay. So I have completed till Codesa Pro, the structure, drawing of chemical structures, and the Codesa Pro, what are the descriptor types it is being generating and all. So now we will see how we can generate files for running Codesa Pro calculation. Okay. So I will start from the structure, like drawing of the structures using Marvin Sketch. So this is the Marvin Sketch platform. So before that, I can show you how to download and how to install the Marvin Sketch. You have to go to the Chemaxon page. So this is the home page of your Marvin Sketch by Chemaxon. So here, if you go to this page, you have an option called download. Okay, you can just click on download option. So now this Chemaxon pass will ask to log in. Okay, if you have already a login ID and password, you can do that, or else we can create an account. Okay, so after creating an account, you will be able to download the software. So you will have to fill up all the relevant details, and then you will be able to download the particular software. Okay, so now I have already downloaded this. I have already registered and downloaded. So now I will show you how to draw a particular chemical structure using Marvin Sketch. Okay. So now if we see, there are different rings are available here, like cyclopentane, cyclohexane, benzene, all kinds of rings are available here. And this is the types of bonds. Okay, so here we have single bond, double bond, triple bond. Then we have the wedges, solid wedges, dash wedges. So everything is available here. So if you go to, like, if you navigate to the different options available, so we can import name, import image. Then if we draw a chemical structure, we can export that as image form, which can be used for the publication purpose and all. We can find the structure online. And for most important tab is structure tab. Okay, so if we draw a chemical structure, we can clean that 2D coordinates. We can clean in 3D coordinates. We can add the explicit hydrogen. And if you're not very familiar, or if you're not confident enough to draw the structures, and if you have the IUPAC name, you can easily generate the structures by giving the IUPAC name. And if you want to generate the name for your chemical compound after drawing, you can click on generate name. So you will get the traditional name as well as the IUPAC name. So if you go to calculations, we can always calculate all these factors like conformers. We can generate the conformers and all by utilizing this Marvin sketch. So I will show you a very simple example how to draw the chemical structures. So if you have the ring, you can just place the ring over here, and we can have the bonds. Okay, this is the chemical bonds. So here, if you have to click at the particular position, and then you have to drag it. Okay, and if you add the functional group, this is the methyl group. Just click your cursor here, and then type in your keyboard. This is an OH group. This is an hydroxyl group. Then again, I am adding putting another ring over here, and then I'm adding the bond. Okay, that is a conjugated pi electron cloud, and then I'm adding certain other functional groups. You can add a fluorine here. So I'm just showing a demo. So if you have a particular chemical structure, you can try drawing it. Okay, so this I have drawn. So now this is in the 2D form, right? So now we can convert this 2D into 3D. So how you will do that? You have to go to the structure tab. Then you have to add explicit hydrogen. Okay, that is the hydrogen. We know that this is a CH2 group, right? So when we are adding explicit hydrogen, this it will show up. That is, hydrogens will be showing up. And after that, we will clean in 3D. Okay, that means the compound in 2D we have cleaned in the 3D. Okay, so this is how you will convert from 2D to 3D. And I will show you how to generate the structure name. We'll remove the explicit hydrogen, go to the structure tab, and then click on generate name. So if you click on generate name, you will have options like preferred IUPAC name, traditional name, CAS number, and all. So if you click on these two, you will get the preferred IUPAC name as well as traditional name. So the traditional name of this compound is A-fluoro 6-methyl naphthalene 1 all, and IUPAC name is 8-fluoro 6-methyl naphthalene 1 all. It is same only. So this is how you can do that. So this is the way for doing or for drawing the chemical structures in Marvin sketch. So now
Ambly, I want to comment something here. See, I want to clarify things to all the participants. Marvin, to some extent, only some options are free for academics, not all the options. And for academics, of course, you have to write to Chemaxon, and they validate whether to give you the license or not. So please don't think that is an openly available software. You have to apply it. That's where Ambli has clearly mentioned you have to create an account, and when you give the details, and they decide whether to give it access to you or not, based on the data you have given. And conformational analysis, many more, as Ambli showed you, like importing images, they works only for the paid version, not for the free version. So the free version, as Ambli rightly said, you can draw structures, save it as different formats, a nominal way of converting 2D to 3D, but she will be okay. There was some issue, but now it is fixed. So I am. She'll be continuing now. Sorry about technical glitch. It is fixed now. So okay. So I hope you are able to see my screen,
I
and
will
here it
show
goes.
the QSAR model generation for anti-hydrogen. So I have already identified the literatures where they have reported anti-hydrogen. Okay, this is the first paper. So in this first paper, they have mentioned about the thiosemi-carbazone derivatives. So if you go down to this paper, you can see this is the structure. Okay, and and there are many compounds are there. They are given the R groups. So similarly, like whatever I have shown you in the marble sketch, you will have to draw the chemical structures by utilizing the rings and bonds. Okay, and the IC50 values are also given here. Okay, so the next paper which I have checked also reported the anti-tyrosine activity. Okay, here if you go down, you can see the structure. So this is the parent structure, and this is the IC50 value which is expressed in micromolar units. So I'm going to merge these two papers, compounds from these two papers because they are characterized by similar bioassay and they have the similar IC50 values. Okay, so I have already drawn the structures for all the chemical compounds in this paper as well as in this paper. So I'm clear that you are able to follow up and you know how to draw the chemical structures, right? So after drawing the chemical structures, here I have shown you how to make the 3D confirmation, right? So CODESA Pro software, one of the most important factor here to be considered is that CODESA Pro will only take the 3D coordinates of chemical molecules. So whatever chemical molecules you are drawing, you will have to convert it into 3D coordinates. Okay, that is the first important thing to be considered for running a CODESA Pro calculation. And the second one is that the naming of your molecule. Okay, so here I'll show you again. We are adding explicit hydrogen clean in 3D. Okay, now we'll have to save this molecule. Go to File, Save As, and we have to save it in the .mol format. Okay, so here you will have to give the file name. So the file name for the structures or the chemical compounds for running the Codesaflow calculation is should be in an alphanumeric combination. That is, there should be an alphabet and there should be the numerical values, and the total length of your file name should be 8 digits. Okay, so I'll give S8 1. Okay, this will be for the first compound. So there is S stands for structure, and rest of all 8001 I have given it for the sake of making it in the 8 digit. Okay, then you can click on save as. Okay, you can navigate to the particular folder that you are having, and then you can click on save as option. So you will have to draw all your chemical compounds and save it in this format. Okay. So I have shown you that I have taken two papers, right? So all the chemical compounds which I have in the first paper, okay, I'll be naming it as S8, then 1 to whatever molecule, whatever number it is there. And second paper, I will name like S9 1 to this much. So when you see the data set or when you see open the folder, you can easily identify that from 1 to 32 is the molecules that is identified from a particular paper, and from S9 1 to whatever 37 or 38 is got from the other paper. Okay, that is to distinguish between the data sets, distinguish between the chemical structures identified from different papers. So I have already drawn these structures, so I will show you. Okay, so I have saved it in the now. I have already a folder created for that. So this is the search for the. So here I have created a folder for moles. Okay. So as I have mentioned, it is an alphanumeric combination. Total is 8 digits. So from S81 to S832. These are the molecules which I have taken from the first paper. And S91 to S937 is from the second paper. Okay. All these are in the 3D coordinates. I have already shown you how to make the 3D coordinates out of the 2D coordinates. Right? Okay. Then second attribute or second important factor is the activity file. How you will make an activity file. Okay. So activity file is I'll show you the activity file. It is an Excel sheet. So this is the particular format for the activity file. Okay, you have to give the title as compound and log IC50. Just leave a space here, and then here you need not give the alphabets. Okay, it should be 7 digits. The ID identifier of chemical compounds should be of 7 digits, and whatever IC50 values are reported in your paper, you will have to convert it into logarithmic form. Okay, so if you see this particular paper, the IC50 is in micromolar. Okay, so this 19.16 will be converting it into logarithmic form. Okay, and then you can you can save it as .txt file or whatever file you want. It should be in the Excel. So now I will again open this activity file which I have created in the Excel. Okay, I'm going to I'm going to copy these values into a notepad. Okay, so here you just click paste. Okay, so now what you can do is that you just give file save as, and in the same folder, like in the F, I had project folder, in QSAR folder, Codesa Pro, in demo. So in the same folder, we are going to rename the activity file. Okay. So in Codesa Pro, the activity file is called as property file. So we will name this as P. Okay. P, which is followed by 7 digits. Okay. So this is the identifier which we will be giving for the activity file. So this is just to make files acceptable for the Codesa Pro calculation. Okay, this should be followed by an extension PRP. Okay, that is nothing but the property. So here instead of text, you just change it to all files. So now you just save it. Okay, so now we'll close all this. Okay, so now it is a Codesa Pro property file. Okay, so now we don't need this activity file anymore, so just delete it. So now we have two attributes, that is our structures and the response values or the IC50 values in the Codesa Pro acceptable format. Now we will be going for the Codesa Pro calculation. Okay, so I have the software here, so I just click it. Now here you have to go to the option, okay, option storage, and you will have to navigate to that particular software. So I have already given the path. If you are not given the path, you just go to browse and then you can give the path. Just click OK. Okay. Now if you go to that particular, you can just close it. If you go to that particular directory where the demo QSAR goes there, all these files are being generated there. Okay, so inside the moles, we have all the 69 chemical compounds that we have drawn, and we will have to shift this Codesa Pro property file to the folder that is props. Okay, now the props is also ready, and we have all the molecular structures available here. Now again, you will have to open the Codesa Pro software. Just click on option storage. Just see whether you are still in the same folder, that is the demo QSR. So just cancel it. And next you have to give calculate load storage. Okay. Now We had 69 chemical compounds in our moles folder, and we had our property file in the props folder. So now what this Codesa Pro software is doing is that they will be getting, or they will be taking each of these molecules, and they will be performing the geometry optimization. Okay, so the geometry optimization protocol which is followed by Codesa Pro is nothing but the MOPAC. Okay, so if you go into detail, MOPAC is a molecular orbital package, and this employs Baker's Eigen following method. Okay, this means that the algorithm will start with an approximation of desired geometry of chemical compound, and then it will calculate the forces acting on the system. Okay, so it will calculate the forces acting on the system until and unless the energy is coming to an optimal minimum. Okay, this is how the MOPAC is doing the calculation. So this Codesa Pro, what it will do is it will take the structures, it will do the MOPAC calculation, and it will generate the geometry optimized coordinates of all these 69 molecules. Okay, and apart from the geometry optimization, the software will also calculate the descriptors. So it will take a considerable amount of time to do this job. Okay, so once this calculation is done and once everything is proper and perfect, you can see here now the structures is shown as 0, descriptor 0, property 0. And now, like, once every calculation is done, once every structure is perfect, you can see the exact number of structures here, the number of descriptors, the property file will be shown as 1 and all. So we will be seeing that, but it will take considerable amount of time. Okay, once this calculation is done, we can go to calculate. Okay, and we can click on BMLR. It is not active now because the calculation is going on. Okay, if you just click on BMLR, this will give you a rough idea that how your QSAR model will be. Okay, it will give the parameters, statistical parameters like thickness, the cross validated R square value, the standard error of estimate and all. But this is not adequate for concluding that your model is reliable and predictable. We'll have to see so many statistical parameters, and we'll have to see the applicability domain of your QSAR model. But this will give you a rough estimate of how the model will be, and we will get the descriptors as well as the optimized coordinates for the molecules. Okay. Now this will take so much of time. So I have already done the calculation. So I will show you how the end result will be. Okay, so I'll just close it. Okay, again I will open. So now we'll have to change the path. Okay, root folder. So now I have to go to browse. So it was on local disk F. Project. QSAR, Codesa Pro, and here I have a particular folder. So demo completed. Okay, just click OK, and then again you click on calculate load storage. Okay, so since all the calculations are being done here, it is showing the number of structures. You remember that we had 69 chemical compounds in our data set. So it is showing 69 structures. The total descriptors calculated is 753. The properties is 1 because we have given one prop file, that is P500, that one that is showing as 1. Okay, it is showing 10 correlations also. I've told you that once this structures, once Codesa Pro optimize all the geometries, it calculates all the descriptors and all, we have to click on calculate BMLR. Okay, if we click on calculate BMLR, we can identify the correlation. That is, the software will tell you the linear relationship between the calculated descriptors and the uploaded property value. Okay, now if we CODESA Pro calculation was to get the descriptor values and to get the optimized 3D coordinates. Okay, that is being done, but this will also give you a rough estimate on the goodness of your QSR model. So I'll explain about each of these terms, what is R square, CV, F, S2 and all during the QSARINS calculation. Okay, so now everything is being done. Okay, the descriptors are being calculated. Now we will check how this we can get this descriptor values. Okay, so go to Codesa Pro. We will navigate to that particular folder where we have completed the calculation. So this is completed, right? So if you go to this particular folder, you can see a particular folder called Mols 3D Mod. Okay, just click there. So we can get all the 69 chemical compounds which are optimized. Okay, optimized coordinates of your chemical compounds can be seen in this Mol 3D Mock folder. Okay, this is very important for the PaDEL calculation. I'll tell you while doing the PaDEL calculation. Now, from these folders where we will get the descriptors. Okay, just click on the folder work and click on this match. Okay, it will be in the .txt format. So you will have to open it with Excel. So now you can see these many descriptors are generated. I have already told you there will be large number of descriptors generated after performing a descriptor calculation on a particular software. Okay, so the end is the R response value or the bioactivity value. So this is not in the number format, right? So you can just select all this. Okay, and then you can change the format. Okay, format says number. So here we can give up to which decimal places we have to keep the data and then just click OK. So now it is in the number format. Okay, we have got the descriptors from Codesa Pro for all the 69 chemical compounds. Okay, so this is how the Codesa Pro calculation is being done. Okay, so the next is our PaDEL calculation. Okay, you just save it. Then we will be moving for the PaDEL descriptor calculation. Okay, so Codesa Pro is done. So PaDEL is a tool. It is a free tool, okay, available for the commercial as well as academic purposes. We can easily download the software and do the calculation. So I will show you how to navigate to that particular web page of PaDEL and how to download the software. Okay. So this is the home page for the PaDEL descriptor. So this software currently calculates 1875 descriptors. Okay, it includes 1D, 2D and 3D descriptors as well as the 12 types of fingerprints also. So how we will download this? Okay, here we have a graphical user interface and Java is very important for running PaDEL calculation. You can easily download the software from here. So it will be downloaded in the form of a zipped file format. Okay, so you will have a zipped file format of your PaDEL. So it will take some time for downloading the PaDEL descriptor. I have already downloaded, so I'll show you how to extract the components and how to run it. So if you go to the softwares, I have already downloaded. It was like this PaDEL descriptor. It is a zipped file format. I have extracted this particular folder, and after extracting, you can see these many number of items will be there. So this is the descriptors. Okay, if you click on this particular thing, you can see what all types of descriptors are being calculated by PaDEL. Okay, the reference is also given, the class of the descriptors, whether it is 2D, 3D, or all every information will be given in this particular folder. So just close it. And if you want to launch this PaDEL descriptor, just click on this executable jar file. So you will get an interface like this. It will take some time. Okay. So now this is the window of our PaDEL descriptor. So now we will have to navigate to the molecules directory file. Okay. So here I will be using the MOPAC optimized 3D geometry of chemical compounds, which was done by the Codesa Pro. Okay. So now I'll go to my folder, that is our demo completed folder, and I'll take that MOLS 3D mock folder where the optimized 3D coordinates of the structure was there. Okay. So go to QSAR, there is a row, and just search. Then so here, if you go, you have the MOS 3D mock file which has the optimized 3D coordinates. Okay. So for simplicity, what I'll do is I'll just copy this folder and I'll paste it in my desktop. Okay. Okay. Now we have the PaDEL, right? So just click here, click on this box. It will take some time. Now we'll have our MOLS 3D mock folder in the desktop, right? So just go to desktop, then click on MOLS 3D mock folder. Okay, you can select this and we can give the path for getting our descriptor output file. For that also you just click on this thing and I'll just save it in the desktop. Okay, that is output file output, and it will be in the CSV format, comma separated file format. Just click on save. And after that, we'll have to choose what kinds of descriptors we'll have to generate, okay? Whether it is 1D or 2D. Then 3D descriptors, if we need, we can just click here. I don't need 3D descriptors right now, so I'm not checking it, okay? And these standardization parameters are very important. So if is there any salt forms in your chemical compound, we will have to remove it, okay? Just click on remove salt and detect aromaticity. That is also very important. And if there is any tautomer-like structures, you can check this. If you think you have to standardize the tautomers, okay. Similarly, standardize the nitro groups, and this is very important. You should check this box, that is retain 3D coordinates. Why? Because already the Codesa software has calculated the geometry or they have done the geometry optimization, right? The software has done the geometry optimization and we are having 3D coordinates. So the same confirmation of chemical compounds should be there, okay? So the confirmation of chemical compounds should be preserved for running the PaDEL calculation also. So we should check it. This is very important. And convert to 3D option, we can click no, or we can give no because already we have completed that calculation. Okay, so just click on start. So now the calculation is being done, and once the calculation is completed, we will get the descriptor output file in our desktop. So here I have given the desktop as a path. If you want to give into any other folder, you can give the specified folder and you can get the output. But remember that output will be in the CSV format, that is a comma separated file format. So now the descriptor calculation is completed in 20.967 seconds and average speed is 0.03 seconds per mole. Okay. Now, if I go to my desktop, so I have given the output, right? Just open this output. So now, here you can see this autogen like value, or this will be given in front of your ID, that is the identifier which you have given for your chemical compounds, you can just remove it. So you just copy this and click Control F, then just click on replace all. So now the 69 replacements has been done. Okay. So regarding with this PaDEL calculation, so these many descriptors, if you see, these are the large pool of descriptors which you have got for the PaDEL calculation. Okay. But if you see this particular Excel sheet, you can see descriptors which are having zero values or the null values. Okay. So if you're finding the descriptors with zero values or null values, you will have to delete it. Okay. You can delete it manually. Okay. You can go to each of this thing, and when you are seeing a zero values, you can delete it manually, because ultimately what we are going to do in a QSR model, it is a statistical approach, it is a mathematical modeling approach, but the ultimate thing is we will have to correlate the activity, the response activity or the biological activity with our descriptors or the structural features. So if their values is zero or null means we won't be able to give a proper correlation, right? So in order to avoid that, in the end, we will have to delete all the descriptors which are having zero values. Okay. So you will have to do it all the like manually you will have to do it. And now we have two sets of descriptors, right? The one which we have done for the by using CODESA, and the one which we have done by using PaDEL. So, by for executing QSAR studies, you can have different combination of descriptors. So, what I'm going to do is that I'm going to merge my descriptors obtained from CODESA Pro as well as PaDEL, and then I will be going forward with the QSAR modeling. Okay, so after deleting all the zero values, you will have to merge the descriptors obtained from CODESA as well as that of the PaDEL. Okay, so it will take some time. So I have already done it. I will show you the end result. Okay, now I have to go to QSAR. Okay. So this is the merged file. So if you see here, you can see the descriptors. The descriptors starting from D00 is the ID of the descriptors calculated by CODESA Pro. And now if you go further, you can see the descriptors calculated by the PaDEL. So I have merged both the descriptors, that is calculated from CODESA Pro and calculated from this particular PaDEL software. So at the end, you will have the response value, that is P5, whatever name property file which we have given, right? That particular identifier was there. The same identifier we can use here. Okay. So this is the form. Now this Excel sheet, we can save it as either in the TXT, that is a text file, or the comma separated file. Okay, we can go to save as, then it is already in the comma separated format. Okay, so now we have generated descriptors from two different sources. We have merged it. We have the values of descriptors as well as the response variables in the CSV format. Okay, so next step is QSAR model building. Okay, so we'll go for the QSAR model building by utilizing the tool that is QSARINS. Okay. So QSARINS is a software developed by the research unit from University of Insubria. We will see how the installation registration process will go on. Okay, I'll help you to navigate to that particular web page. So this is the web page for the QSARINS software. Okay. So now, if you need to get this license of this particular software, you will have to go with the registration process. Okay. So just click on this particular tab. So now, before doing QSARINS, I would recommend to read read the protocol. Okay, protocol, it's not the protocol, it's a manual. So you will have to read the manual. There are so many statistical parameters. You will have to understand what is the statistical parameters. Even though if you're not able to understand the equations and all, you will have to understand what is the significance of those statistical parameters, and then you will have to understand what are the cutoff values. Okay, cutoff values which are implemented for these all statistical parameters according to the OECD principles. Okay, so OECD principles are very important for QSAR model development. According to OECD principles, a QSAR model is said to be reliable or it is said to be predictable if it is satisfying certain conditions. Okay, like the conditions is given for the statistical parameters. If your QSAR model is having the statistical parameters within the cutoff value, we can say that that QSAR model is reliable, it is predictable, and it is reproducible. I'm sorry. And now here we have to click the register. So if you are a registered user, you can just click on login. You will get a user ID and password and you can log in into that particular system. But here if you are using it first time, you can click here to register. So one of the most important things is that only the people with fixed position in academia or research center will obtain licenses. So you won't be able to download the software. Student research scholars or students won't be able to download the software, but your group leader can register. So you can give in all the details here. You have to give the details like how did you know about QSARINS, what is the purpose, what is your experience in QSAR model building, and if you have any relevant publications related to QSAR, you will have to give it. Okay, and you will have to complete the the registration process. So once you complete the registration process, you will get the license, that is a license for using that particular software QSARINS. Once you got it, you can go with the QSAR modeling. Okay, now I will show you what is how the interface of QSARINS will look like. So, sorry. So now this is the particular interface for the QSAR model. Okay, I mean QSARIN software. So now how will you do a calculation here in QSARINS? That is the very important step. Now if you go to the particular tabs, okay, you can see file, you can see analysis, tools, as well as you can see the help. So we'll see what all these things are. So if you just need to import your data, so data in the sense I have already shown you the merged file, right? The merged file for the descriptors from CODESA as well as PaDEL along with its activity values, right? So this data we'll have to import. So you'll have to remember that this software will take only txt file or .csv file for the data. So we have already saved it in the CSV format. So we'll go to import data. Now we'll have to navigate ourselves to that particular folder. Go to project, go to QSAR, go to table, and I'm adding demo. The demo merged. This file you will have to open. So now, since it is in the comma separated file format, the delimiter you will have to give it as home. Just put a comma in that box. If it is a text image, you can just check this particular tab. So now, this is very important. These two parameters are very, very important. We will have to check these two boxes. Okay, just click yes. Here also, just check it. So this states that we will have to delete or we will have to remove all the descriptors which are having correlation greater than 95% and the constant greater than 80%. So what is the significance of doing this? In QSAR model building, we have a particular concept called orthogonal descriptors. So orthogonal descriptors are nothing but they are highly intercorrelated descriptors. Okay. So by checking these two boxes, we are telling the software to remove highly intercorrelated descriptors. Okay. So why we have to do this? Because as I have mentioned earlier, the ultimate aim of QSR model building is to relate our descriptors with our activity. It can be biological, physical, or chemical activity. So if we are explaining our biological activity with the intercorrelated descriptors, what is the meaning or what is the significance of doing that? There is no significance. So we'll have to remove all those intercorrelated descriptors. Okay. So after checking these two boxes, just click on import. Just click yes. Now it is importing by filtering the correlated descriptors. Okay, it will take some time. So we will have to see the output once this calculation is being done. So now it is filtering the correlated descriptors among the among the one which we have done for Codesa Pro as well as the PaDEL. Okay, there will be so many correlated descriptors. So now they are filtering all the correlated descriptors. Okay. So now there we can see total excluded descriptors are 693. Okay. So these many descriptors were highly intercorrelated. Okay. All these descriptors are removed by the software. So the next step is just click on the tab analysis and data set. Okay. Now if you go here, you can see these are the descriptors. Okay. The descriptors starting with D00, they are the descriptors calculated by Codesa Pro, and the descriptors starting with the A log P are the ones which have been calculated by the PaDEL. And the last one, P5001, is our biological activity value, that is a response variable. Okay. Now, what we have to do is we have to select the variables, that is the descriptor values, and then we have to select the response. We have to do it manually. Just click on the like one button and then press shift. We'll have to do this manually by pressing the shift button. You can just select all the descriptors. So here is your descriptor. So now we'll just click on select variable. Okay. So now all these descriptors are being selected. Now just click on this particular tab, and this is our response value. Okay, that was in the CSV format. Then just click on select response. Now we have all the data which is required for our model. We have the descriptor values and we have the response variables. Okay. Then the next very important step is the dataset splitting. Okay. So the dataset splitting means we will have to divide the total number of compounds in our dataset into training as well as test set. Okay. So how will you do that? So how will you determine whether these compounds should go to the training set and this compound should go with the test set? Okay, we have different approaches for doing that. The first one is for every nth selection. Okay, that means I'll just tell you an example. If there are 30 compounds in your data set, okay, so 30 compounds are there in your data set, and the training set compounds to test set compounds ratio is 4 is to 1. Okay, so you will have to split the entire data set into training set and test set in the ratio 4 is to 1. So if you do so, the starting from the first compound, every fifth compound will go to the test set. Okay, the remaining will go to the training set. Okay, so that is the every end selection criteria. Okay, the next we can go with the random percentage. We can usually we use 70 and 30 splitting. That means 70 percentage compounds in the training set and the rest 30 percentage in the test set. Okay, so here if we give the random percentage, okay, so here you can give like 30. So 30 will be the prediction set or the test set, and others will be the training set. And another option is based on the clustering studies. So I would like to recall Dr. Giri's presentation on star drop. So he has mentioned about the clustering approach that is being employed in star drop, and this after performing this clustering, we will be able to identify certain unique set of compounds and the rest of compounds which are sharing similar scaffolds. Okay, so this unique set of compounds which are not sharing the similar scaffolds can be treated in the test set and others in the training set because the model building is entirely depending upon the training set. The test set is also having their importance, so we will have to see all these approaches, like whether we have to go for a random approach, whether we have to go for an end selection. So for end selection, when you are doing, you will have to arrange your activity values in the ascending order first. Okay. Then by using a 4:1 ratio splitting, starting from the first compound, you can include every fifth compound in the test set or in the prediction set, or whether you have to go with the clustering analysis. Okay, so this is how the data set splitting is done. The splitting is a very important criteria. So if you change the splitting parameters or the splitting parameters, what it is implying is that there will be drastic changes in the statistical parameters of the QSAR model generated. Okay. Now, if you are done with selection of your variables, selection of your responses, as well as the training as well as prediction set classification, you can just click on OK and again go to analysis, then go to the variable selection and models calculation. Okay, so here the algorithm employed is GA. Just check on GA, and here the iterations we can choose whether we have to go with the 500 iteration or whether we have to go with the 10,000 iteration. Okay, so I would recommend everyone to go with larger number of iterations because as we go with a larger number of iterations, we will be able to get the most significant descriptor which could be correlated with the biological activity. Okay, so the other parameters we can keep it as default, that is the mutation rate, population size, and all. So, as I have mentioned in the very first slide, GA is based on the evolutionary algorithm. So, it will have all the biological parameters. The naming is given in that sense. So, the mutation rate and all means the different combinations of descriptors will be identified and the most correlated descriptors will be chosen for a particular QSAR model. So we can keep all this as a default parameter and then we just click on calculate. Okay, so now it is calculating for two descriptors. So once this two descriptor calculation is done for the 10,000 iteration, again you will have to click on the analysis tab. Again you have to click on variable selection and you have to repeat it for the three descriptors, then four descriptors. So similarly you have to go with that. Now once this is being done, okay, so this will take so much of considerable amount of time. So I have already calculated this QSAR model, so I can show the QSAR model and the statistical parameters associated with this. So I'll just okay. Now I'll once this calculation is being done, okay, once you are done with your model, once you are done with your statistical features, you just click on File, Save Project. Okay, the project will be saved as .qsi format. Now I'll open the project file which I have saved already. That is in the local disk file. QSAR folder. Yes. Okay. So I have saved it as inside this folder. This is yes. Now the calculation is being done and I have saved the project file. Now I will show you the models. Okay. So these are the models generated during this QSAR model. Okay. Model development. So we have the descriptors like one descriptor, modules with one descriptors are there, two descriptors are there. So I have done till the five descriptors. So a very good QSAR model is the one having minimum number of descriptor values. Okay, so we should always check for a QSAR model which is having very less number of descriptor values. Okay, so I have selected the model which is having three descriptors. So how you will select this model, you will unselect and all, you have different options available here. So for selecting, you will just have to click on the model and click on this S. So the model will be selected. So to unselect it, you will just have to select it, just click unselect. Okay. So all these models I'm just unselecting. So this is the model which I have got, and this model has got very good statistical features compared to all other models in this QSAR model development. Okay, so I have checked the statistical parameters associated with each of this model, like the one descriptor model, two descriptor model, and three descriptor model. Up to five descriptors I have done, and at last I identified that this model, that is with the ID 3712, has got very good statistical features. Okay, statistical parameters as well as that of the biological significance with respect to my activity, because my activity which I have chosen was anti-tyrosinase activity, right? So now I will select this model and you will have to perform the validation. Here you can see the R square is 0.82. I'll be explaining each of these parameters. So now you will have to validate it. So this is for the internal validation. Okay. So just click this and it will perform the internal validation. I'll explain what all these parameters are. So internal validation is nothing but during internal validation, we will be validating our model by using the data which we have created the model. Okay, that is called internal validation. And during external validation, we'll be having a totally external set of chemical compounds and we will be analyzing the predictive ability of QSAR model. Okay, now we have done with the internal validation and this is for the external validation. There are certain parameters reflecting the external validation. So that calculation is also being done. Now, if we open this particular window, so there are fitting criteria, then there are internal validation criteria, external validation criteria, and some other statistical parameters, and so many graphs are available here. So we'll see what all these parameters depict. Okay. So in the fitting criteria, we have R square. So this is a model for 3 descriptor and the coefficients are given here and the standard error also. So we will need the descriptors which are having less standard error and the p-value should be also less for the statistical significance. Okay, the p-value should be less, standard error should be less, and the correlation, that is the intercorrelation between the descriptors should not be there. So if we need to check how much intercorrelation is there with between these descriptors, we can just go to this particular icon, and this is a correlation matrix. So if you click on this correlation matrix, you can see if there is yellow color, it is showing 70% correlation. Orange color means 80% correlation, and here red means 90% correlation. But we could not see any particular color patterns between these descriptors. So this signifies the fact that these descriptors are not intercorrelated. Okay, so this is the correlation matrix. Now, if we check the fitting criteria, one of the most important statistical parameter is the R square. Okay, that is a regression coefficient. So this R square value, it helps to evaluate the fitness of a particular model. Okay, so usually it is said that the value of R square closer to zero. This means that the model is good, but we cannot say the model is good only based on the value of R square. We have to see different other parameters like the internal validation parameters, external validation parameters, and so on. But generally, R square value greater than zero point 0.6 is acceptable for the QSAR model generation. And the next is R square edges. Okay, so this parameter, okay, this parameter is also very important because as we increase the number of descriptors, okay, as we are going to increase the number of descriptors, we will get a very good R square. Okay, but this will not be statistically good, or it will not be statistically relevant. Okay, so adjacent R square values are also calculated to avoid this particular statistical incompatibility. Okay, then LOF is the lack of fit score. Okay, so this LOF score is a measure of error. So this should be close to zero, or it should not be greater than zero point 0.5. That is the LOF criteria. And we have some other parameters also, like we have parameters like KXX, RMSE, MAE, S value, concordance correlation coefficient. Then RSS stands for residual sum of squares, and F is the Fisher criteria. Okay, so I'll explain each of these parameters. So the error of estimate, or the error values, are usually depicted by utilizing the terminologies like RMSE training, MAE training. Then we have another parameters which is related to that LOF value. So these all is related to the error value. So these error values should be less. It should not be very high. Okay, that is one of the most important thing to be considered. And the next is when we are considering about the F value. Okay, F value is the Fisher criteria. So higher values of F implies that the model which we got is statistically significant, as well as the model is not obtained by chance correlation. Okay, so always we have to see for the higher F values for getting a statistically significant QSAR model. Okay, and we have some other parameters are also available to this particular thing. I've explained about LOF. Next is KXX. So what is this KXX? It is the global correlation among descriptors. Okay, so the global correlation among descriptors also, it is showing somewhat low value, only 0.30 it is showing. And MAE stands for mean absolute error. Okay, so mean absolute error values are also less. Okay, CCC training is concordance correlation coefficient. Okay, that is showing 0.90. It should be high, it should be close to 1. So the fitting criteria, if we check, the model is having a good fitting criteria. Okay, and when we are saying about the RMSE value, usually the RMSE value, when we are checking the RMSE of training set, then RMSE of validation set, and the S value. Okay, that is the overall error related to that particular model. Should have values which are closer. It should not be very high. It should have the closer values. So now, if we see the S value and RMSE training, it is having the closer values. Now, next, moving on to the internal validation criteria. So, as I have told, the internal validation is performed using the data which created the model, right? So, we have certain statistical parameters, okay? So, here you can see Q square, that is nothing but cross validated R square. Q square is nothing but cross validated R square. So for the cross validation, we are employing two different methodologies. That is leave one out method and leave many out method. Okay, that is in short form it is given as LOO and LMO. So how this leave one out method is performed? So we will be having a fixed number of molecules and fixed number of descriptors. But when we are performing a LU calculation, that is leave one out calculation, the model will be built by considering experimental property of n minus 1 compounds. That is, one compound will be omitted and their respective descriptors will be also omitted. Now we will get a model, right, with the n-1 chemical compounds. This model will be used to make the prediction for the left out compound. So similarly, leave many out method means many molecules will be omitted at a time and their respective descriptors will be omitted at a time, and the model which we get without that many molecules will be used to make the predictions for the left out molecules. So these are the two internal validation criteria, that is LU as well as LMO. Now, if we see the Q square U value, okay, it should be also greater than zero point 0.6. Okay, so if the Q square U and LMO values are greater than zero point 0.6, you can say that the validation, you have got a very good statistically significant model. Okay, and also we have another parameters like RMSE-CV, then MAE-CV, and also these values should be less. Okay, should be less, but there is one of the most important criteria is there, that is the RMSE training should be less than RMSE CV. And I have already told that all the RMSE values like RMSE of training, RMSE CV, and the standard error of estimate should have the values which are closer. So if we see here, the RMSE CV value is coming as 0.56 and RMSE training as 0.50. So the RMSE training has a lesser value compared to RMSE CV. This is one of the most important criteria for getting a reliable QSAR model. Okay, and another internal validation method employed by QSARINS is nothing but the randomization. So how this process of randomization is being done? Okay, or what is the significance of randomization? So randomization is being done by shuffling the activity values. Okay, so we will be getting the activity values from the literature, right? So we will model the chemical compounds by using their respective activity values, but during the randomization process what happens is that the activity values of chemical compounds will be randomly shuffled and the model will be created. Okay, so now you will have to think whether you should get a very good R square values for this randomization process. So the process which we say here is a Y scrambling. Okay, so we are also identifying the R square Y scrambling as well as Q square Y scrambling. So we should have or we should get lower values for this R square Y scrambling as well as Q square Y scrambling, because if you are getting higher values for these two parameters, that is R square and Q square, the model doesn't make any sense, because whatever bioactivity value we have given for a particular compound, it should be specific for that particular compound. If we are shuffling the activity value, and again, if we are getting a good model with a very good R square and a very good Q square, means our model is bad, because the specificity is not there. Okay, so the values of R square Y scrambling and Q square Y scrambling should be less. So if you can see here, you can say that the value of R square Y scrambling is 0.06, which is very less, and Q square Y scrambling came into negative value, that is minus 0.1188. So we can say that the model which we have created is not obtained by a chance correlation. It is a well developed or a statistically significant QSAR model only. Okay, so this is the internal validation criteria. So, several scientists have discovered the parameters like R square external, Q square F1, Q square F2, Q square F3, then concordance correlation coefficient external, R square and average and all to evaluate the external validation criteria. There are cutoffs for these values. The Q square F1, F2, F3 values should be greater than zero point 0.7 for getting a statistically significant QSAR model. Then the concordance correlation coefficient should be above 0.85. The R square external value should be greater than zero point 0.6. R square M that was introduced by Dr. Kunal Roy and it should be greater than zero point 0.6. Q square F1, F2, F3 were proposed by Tropsha and the RMSE external should be very less. Okay, it should be less but comparable with the RMSE and the overall error of our model. Okay, so now if we see all the criteria which this model is meeting all the criteria which I have mentioned. Okay, and now if we go down, we can get two other parameters K and K dash. Okay, what is this K and K dash? Okay, these are the slopes of our regression line. I have already told you that in QSARINS they are employing MLR based approach, right? So it will help us in deriving a linear relationship between the independent and dependent variables. Okay, so the slopes of the regression line are depicted by K and K dash. Okay, so the value of K and K dash should be between 0.85 and 1.15. So if you see this, you can easily identify it should be between or equal to 0.85 and 1.15. Okay, so we can say that we have discussed a large number of statistical parameters and their cutoff values. Okay, so this model is satisfying all the cutoff values and all the parameters, all the statistically significant parameters we have mentioned, and it is above the cutoff values. Now we'll have to see certain graphical representation. Okay, so here we will have the scatter plot. So this is the scatter plot which is being done with the experimental endpoint and the predicted by the model equation, because after the QSAR model generation, the model equation will be generated by using the descriptor coefficients. So by utilizing those predicted values and the experimental endpoint, the scatter plot has been constructed. So in scatter plot, we can see that there is a uniform distribution of the data points. Okay, so the yellow color indicates the compounds in training set, and the blue color indicates the compounds in the prediction set. Similarly, if you go here, it is also the scatter plot which is being calculated by the that is leave one out method. So here also we can see a uniform distribution of your data points. Okay. The next is the residual plot. What is the meaning of residual? Okay, residual means residuals are nothing but the difference between experimental and predicted values are called as residual values. Now we'll have a plot of experimental endpoint and the residual value. So this is how the graph is representing. Here also we have the uniform distribution with the line passing through the zero or the with the line passing through the zero. So it is showing a very good prediction only. And this is Williams plot. Okay, Williams plot help us to identify the applicability domain of your QSAR model. Okay, so for that we will have to check what is the applicability domain. Okay, what is the significance of applicability domain? So assessment of applicability domain is important to identify whether the model is able to make predictions within the chemical space. Okay, so the main approach employed for this is the leverage based approach. Okay, so if there is any chemical compounds which is having the structural features distinct from the rest of compounds in data set, it will be identified as an outlier. Okay, so here you can see at the top it is showing hat values, right? So the hat value is given as 0.255. Okay, so that is the threshold value H star. So how this H star is being calculated? There's an equation that is 3 into K plus 1 divided by N. So N is the number of training set compounds and K is the number of descriptors used in the model. Okay, so the compounds with leverage less than the threshold value, so here it is 0.255, they are considered to be within the applicability domain and they are reliably predicted by the model. But if you see this, there are three structures which is in the training set. They are considered as the outliers because they are not falling within the applicability domain of QSAR model. So if we remove these compounds, okay, if we remove these outliers, definitely our model will get improved, but we will have to see what is the reason or why these chemical compounds came as outliers or why this is depicted as outliers. If there is any structural features, or we say that this structural feature is responsible for making this compound as outlier. So we'll have to acknowledge that, or we'll have to think in that way, and we'll have to put it in our manuscript. Okay, so this is how the applicability domain plot is there, and this is called Insubria graph. Okay, Insubria graph, it is also a variation of Williams plot for predicting the results of chemicals without experimental values. Here also you can see there are two outliers are there. So if you go to the Williams plot, then you can name it. Okay, if you name it, the compounds like 9, 32, and 26, they are shown as outliers. Okay, similarly, if you go to the plot in Subria graph, here also you can see that 26 and 32 is represented as the outlier. And if the chemical compound are in the outlier region, but if it is closer to the applicability domain, here and now we can see the SA32 structure is very closer to the applicability domain threshold. So it is not a very big deal, but if it is very far, we have to check, or we have to see the reason whether any structural features are responsible for making it as outlier or not. Okay, and this is the LMO plot, that is a Q square LMO and the model Q square. So the model is falling within the limit of the particular descriptor values, and the last one is the bi-scrambling plot. So here you can see the model is very far from all the data points. So this means that the model is not obtained by the means of chance correlation. Okay, it has got the specific bioactivity values for the particular chemical compounds, and the modeling is done based on that. Okay, so this means that the model is not obtained by the means of chance correlation. Okay, so this is how you will have to identify the statistically significant QSAR model. Okay, so till this time I have mentioned about the statistical significance. What are the parameters required for the statistical significance? What are the cutoff values? Everything we have discussed. But the ultimate thing, as I was mentioning from the starting or from the beginning, it is the correlation of your descriptors with the biological activity. Okay, so here if you see, there are three descriptors are there, out of which two descriptors are from CODESA calculation and the third one is from the PaDEL calculation. Now we will have to see what is the significance of these descriptors. Okay, so if you need to check the physical meaning of the descriptors which are calculated from the CODESA probe, what you will have to do is that you will have to navigate to that particular folder. So just go to CODESA Pro and just search for your name. So now, if you just go to this folders, you can identify a particular folder called descriptors or DES series. So just click there. So here you will have the descriptors calculated for each of the chemical compound. Okay. So just open any one of the file. I'm opening the very first file. So here The very first descriptor is D0289 and we have D0364. Okay, so here we'll just find this descriptor 0289. Okay, so this 0289 depicts LUMO energy. Okay, what is LUMO energy? That is lowest unoccupied molecular orbital. Okay, and what is the significance of 364? 0364. It depicts the vibrational enthalpy. So now we have got the importance or physical meaning of these two descriptors. That is 289 corresponds to LUMO energy and 364 corresponds to vibrational enthalpy. So we'll have to see how this could be correlated with our biological activity. So when we are saying about the LUMO energy, so what is a HOMO-LUMO or what is the significance of HOMO-LUMO in chemical bonding? HOMO is highest occupied molecular orbital and LUMO is lowest unoccupied molecular orbital. So if the gap between the HOMO and LUMO is less, the electronic excitation or the ease of electronic excitation will be higher so that chemical bond formation can be made or the bond formation can be made easier, right? So this descriptor is representing the chemical bonding behavior of the compounds present in the dataset. So as we all know that we are dealing with the protein ligand interaction. So after this QSAR, I have already performed the docking studies and all. So the docking studies was done with the target that is tyrosinase. So how will you depict or how will you infer that the docking is proper or you got a reliable result after performing a docking studies? We'll have to check the interaction with amino acids. Obviously, the factor that energy values, the binding affinity values, everything are important. But if the chemical compound is not making significant interactions with the catalytic site of particular protein, or if it is not making significant interactions with the binding site of your protein of interest, whatever docking you have done is waste. It is not going to tell you anything. The biochemical information cannot be extracted from such docking studies. Okay, so the strength of a protein and ligand, okay, it depends on the chemical bond formation. Okay, so the interactions, it can be hydrogen bond interactions, it can be hydrophobic interactions. So this descriptor is accounting for the chemical bonding behavior, okay, behavior of your the compounds present in the data set. So the second descriptor 364 that was related with the vibrational enthalpy. So how you will encounter the vibrations within molecule? We can say that there are stretching bond vibrations are there, bending vibrations are there. There are different types of vibrations are there, right? So this vibrational enthalpy of molecules or the vibrations in the molecules are also correlated with the bonding or it is correlated with the polarizability of the molecules. It is correlated with the structure elucidation of the molecules, right? So if you are having highly polarizable molecule, you cannot see a stretching, right? Because the if you are seeing a highly electronegative molecule, this electronegative atom in a molecule, all the electron cloud will be dominated with that particular atom, and you cannot see the vibrations like much higher frequency of vibrations. If it is not there, there won't be adequate charge separation between this atom and their neighboring atoms. So, if the charge separation is not proper, we cannot account for the chemical bonding, right? Chemical bonding always involves the electrostatic interactions as well as other interactions are also there, but electrostatic interaction is the predominant one. So this vibrational enthalpy also accounts for the bonding potential, the polarizability factors, as well as the structural features of chemical compound. So this is also very significant considering the stability of your protein ligand complex. And the last descriptor, that is SSNH. Okay, so I've shown you in the beginning, if you go to the PaDEL software, there are list of descriptors, right? Just you search for the SSNH descriptor. So this descriptor accounts for the total number of NH groups, okay, NH groups that is present in your data set. So if I go to my data set, okay, this data set mainly consists of carbazone derivatives. So here you can see this NNH double bond is NH2. So this group is permanent for all the molecules. The R group is only changing. So the NH group has got significance. So I have conducted some literature studies also to identify the significance of these NH groups. So it has also mentioned that the presence of electronegative elements like nitrogen, fluorine, and all in this thiosemi carbazone derivatives can increase the potency of these molecules towards their target, that is the tyrosinase. Okay, so now we can see the structure had NH groups, and this NH groups had great role in eliciting the potency of thiosemi carbazone derivatives. So this is reflected in my QSAR model. See, you can see the SSNH groups here. Okay, so if you do a perfect QSAR model, this is it should have certain descriptor values which should correlate with the structure and the potency. So out of the three descriptors, the two of the descriptors were more related to the stability of protein ligand complexes by emphasizing about the chemical bonding potential and all. The last descriptor was clearly mentioning the relationship between the groups present in the chemical compound and their potency. So this is the explanation, like we are relating the physical significance of the descriptor with respect to their potency or the biological activity. Okay, so this is how it is being done. So now we have learned how to do a QSAR model, how to do the descriptor calculation, how to prepare the files, how to do the QSAR model, what all statistical parameters we have to keep in account, and what all graphs we'll have to identify, or what all significant, or how we can correlate the physical significance of the descriptor with the potency. This all we have covered. Okay, now the ultimate aim or the most significant factor here we have to consider is the external validation of QSAR model. Okay, how will you do the external validation? Even though QSARINS predicted some of the external parameters like Q square F1, F2, F3, concordance, correlation coefficient, and all, and this model is satisfying all the criteria, all the cutoff values for these statistical parameters. Still, we will have to do the external validation of QSAR model. How we will do that? Okay. So for doing the external validation, we will have to identify new set of molecules, okay, which were not used for the QSAR model building. So if for this QSAR model building, I have used two different papers, compounds from two different papers, and I have generated the QSAR model. Now, for the external validation, we will have to identify a new set of molecules, okay, which were not used for the QSAR model building, but they should have similar bioassay, like the tyrosinase inhibitory activity was considered for this study. So I have chosen 10 chemical compounds which were reported for the tyrosinase inhibitory activity itself, but that those 10 compounds were not used for my model building. Okay, so for external validation, we'll have to identify those chemical structures. Same procedure we will have to follow. We'll have to draw the chemical structures. We'll have to convert into 3D coordinates. We'll have to do the descriptor calculations. So take descriptor calculation, it is same. We'll have to do the all the steps which I have mentioned for the QSAR model building. Now, after performing the descriptor calculation, we had three descriptors here, right? The 289, 364, and SSNH descriptor. So we will have to identify the values of these descriptors for the external compounds. Okay, we have done the descriptor calculations, right? From the CSV file, we will have to identify what is the values for these three descriptors, because we are going to validate that model by using the coefficients of these descriptor values. So I have already done this. So these were the 10 chemical compounds which I have chosen for doing the external validation. So we have 289, 364 SSNH groups, sorry, SSNH descriptors. So the values of these descriptors were identified after performing a CODESA-approved descriptor calculation for these 10 compounds and the PaDEL calculation for these 10 compounds. Okay, and it should have the IC50 values also. The potency value against tyrosinase was also expressed in that paper. So I have converted the IC50 values into logarithmic values. Okay, and the next factor here that we have to consider is the calculation of PIC50. So how I will calculate the PIC50? So the PIC50 calculation is done by analyzing or by utilizing the coefficients from the model equation. Okay, so here if you go to the QSARINS, you can see the intercept is having 5.12 value. Okay, this descriptor, that is LUMO energy descriptor, is having the coefficient value of minus 1.74. The vibrational enthalpy descriptor has the coefficient value of minus 0.02. The SSNH descriptor has the coefficient value of 0.89. So by utilizing these coefficients and descriptor value, we are going to get the predicted IC50 value. I'll show you how this is being done. So if we just select this PIC50 value for the first compound, this is the equation. Okay, this equation you have seen, right? This is the intercept plus this is the coefficient value, that is coefficient value of the LUMO energy descriptor minus 1.7 into descriptor value of that particular compound for the LUMO energy, plus second one is the coefficient of vibrational enthalpy descriptor into the descriptor value of SH1 chemical compound. Then the third one is SSNH. Okay, the coefficient value is 0.89 multiplied by the descriptor value. Okay, so this mathematical expression will give you the PIC50 value. Once the PIC50 value of the first compound is done, you can just click on here, drag until the end. You will get the PIC50 values of all chemical compounds under consideration. Okay. Okay, so this is how you have to do the calculation. Now if you have you have done this particular calculation, you can plot a scatter plot. Okay, you can have a scatter plot, and then you have to identify the R square value between the PIC50 value and the log IC50 value. Okay, for getting this R square value, what I have done is you can just plot a scatter plot. Just select all these values and then just you can make a scatter plot. You just go to insert, then charts, you can go to scatter plot. It will give you a scatter plot, and if you just click here, it will show a trend line. And then if you want to just show the R square, just click on the linear option, just click on the more option, and then just click on the display R square value on the chart. Okay, so I have already done this calculation. So since I have already done this calculation, I have got the R square value as 0.86. So here we can say that this value, the R square value is 0.86, right? So it is showing that if we have taken the external set of compounds and we have calculated their predicted IC50 using the coefficients of our model, the descriptors of our model, and then we have compared the PIC50, the predicted IC50 value with the log IC50 value, and we have calculated the fitness function. It is giving the value above 0.6. The value is nearly 0.86 for the 10 compounds. So it is stating that this model can be used to predict the activity, that is the IC50, predicted IC50 value of chemical compounds. You will just have to get the intercept values and the coefficient values. So by utilizing that expression, that is intercept plus coefficient into descriptor value, Okay, then plus the coefficient of second descriptor into the corresponding descriptor value will give you the PIC50 value. So this is how you can externally validate. Okay, so this is done by using the manual calculation. So we have seen, so far we have seen about the QSAR model development. We have seen what is the QSAR, what are the important attributes of a QSAR. We have to identify the descriptor values. We have to identify the response values. Then we'll have to go for the QSAR modeling. We'll have to be very clear about each step, and we have to draw the structures. We have to optimize their geometry. We have to do the descriptor calculation, and then finally we have to do the model generation. And we should be very clear about the OECD principles. We should be very clear about the cutoff values for each of the statistically significant parameter, and then only we can say that our model is statistically relevant. But only the statistical significance alone will not suffice our need, because we will have to search, or we'll have to think about we'll have to correlate the significance, physical significance of the descriptors obtained in QSAR model with our biological activity. Then only we can say that our QSAR model is good. Okay, ultimately we will have to do an external validation to validate the QSAR model which we have done, to check whether this QSAR model is able to predict the activity of external set of chemical compounds. So this is the entire procedure for doing a QSAR model. Okay, so if you have any doubts or if you want any clarifications, you can ask me. So this is how it has been done. Thank you.
Okay, so that was an extensive uh, demo presentation. Thank you so much, Ampli. I really appreciate your time and patience on preparing your demo slides, as well as the data that to be taken care. So I really appreciate that. So let me give you a few insights before moving to the questions. As she already mentioned, merging of the descriptors. It is not necessary, but as I told you in my to-do or do's and don'ts of QSR, it is always better to have a good pool of descriptors, which is very desired, focused set of descriptors, not simply some set of descriptors, right? And then try to correlate with your physical meaning or physical significance of your descriptors with your activity, and also statistical significance. So we tried with several combinations and ended up with CODESA or MOPAC derived descriptors, as well as with PaDEL descriptors. And also keep in mind, Ambli was using the MOPAC 3D optimized geometry for PaDEL descriptor generation. If you're using a docked, let's say you did docking and you want to use docked complex poses, take the poses, don't do any optimization, use the same geometry to generate the descriptors. You can do the same way what Ambli did in PaDEL descriptors. Only thing is you're taking the mol files or SDF files from the docked poses, so that you get a biological complex conformer. So you need not to redo a optimization there. So it is all about where you want to apply, how you want to take it. And keep in mind the applicability domain, what she discussed, and also at the end, external validation, completely taking a different data set of the same bioassay from a literature published data with experimental, and then we did a comparative prediction using manually the equation. And also keep in mind we used MLR has overfitting issues, so you have to keep that in mind, so that your models, you have to look at each and every statistical parameter, where Humbly was really emphasizing a lot into the cutoff values, its significance. Otherwise MLR will get into false positives. So be very careful with the method MLR. In order to overcome these overfitting issues, of course, you can use PLS method, partially squash, or Gaussian process regression method, which is again a machine learning method, or even you can use what to say, random forest method, There are different methods available. There are different tools available for that. Anyway, moving forward, Ambli, if you wanted to answer some of the question, please unmute yourself and go ahead. I'll read out the question. If you wanted to answer, please go ahead. So the first question, please send link for QSR. Really, I didn't understand this question, because what do you want? If it was related to literature, I already shared it in the Google Meet as well as in the YouTube links in the in the chat. If it was for the software, please go to YouTube and go through what Ambli was referring to. The websites were already in her slides. Please let me know if there are any other links required. How is QSARINS better than others? As I told you, for MLR, it is considering a lot of statistical parameters that which might lead you to false positives. So, and that way it is good. And QSARINS also reporting or also calculating many statistical parameters, which is very, very helpful for you to make a decision whether your models are good or not. So keep in mind, QSR is cannot be black boxing. If you have any commercial tools, or if you have any other tools, whatever you're using, just a click of a button, you get an R square. Sorry, I have to tell you, each step you need to know what you're really doing it. Software does wonderful job, but you have to have your put your human expertise to have a better outcome or reliability and reproducibility of your QSR models. So for MLR, with a combination of a genetic algorithm and QSARINs, where they do the feature selection, that is a descriptor selection. Hope you have seen there are several pools of descriptors, like 3 descriptors, 4 descriptors with different R square and Q square comes. There are multiple models comes, and then we do several iterations and pick them up. Even though in the demo Ambli did not show several iterations, she has done several hope you saw her folders in her most of the QSR, so that there were a lot of folders that she were doing iterations. So after several iterations only you can come to a conclusion which could be the best model with lower error with the within the cutoff value. And she was also acknowledging some of the parameters from Tropsha as well as from Dr. Kunal Roy. So they were also having some parameterization, the statistical validations. So that way QSARINS is good. Any other softwares for modeling? As I told you, you can use Veka, you can use Statistica, you can use Orange. Orange is another tool for SVM or neural network or random forest. These are all free, what I'm telling. Okay, I'm not referring to any paid softwares, it's all free. Then NIME, I'll be doing a session on NIME for QSAR modeling also. So there are plenty of softwares available for QSAR modeling. How to get your session updates? Any WhatsApp or Telegram group? Okay, I'm going to show you the number so that you can send me a message to that number. I will add it to the group so that you will get to know the sessions. What if I don't have a data set more than 20 compounds? Is it still possible to generate a QSAR model? How reliable the model will be? Ambli, do you want to answer this question? You can unmute yourself and answer if you want. Ambli, are you able to hear me? Oh, Ambli, can you hear me? Oh, so I'm not sure. Oh, she's sending some message. Oh, your audio has some issues. Okay, fine. Let me take this. See, taking 20 compounds is fine if it is homogeneous or if it is a local model. Means you have the exact set of chemical class of compounds with a certain derivatives. They are absolutely fine. But if you are going for a diverse set of molecules or heterogeneous compounds, then 20 is not enough. So earlier days in 90s, the papers were mostly concentrated on local models. That means homogeneous models, and the reliability and reproducibility of QSAR models were much better. Once the software technology and many more multiple options have been given to us, a little bit of exploit. Sorry to use that word. We exploited it. So without knowing the science behind it, we started even I did initially the same mistakes. I don't know about others. I did the mistakes. So I was exploiting it, putting several diverse sets and not looking too much care into the false positives. And of course, I didn't publish anything at that point. I was when I was reporting, and that's where my supervisor is able to correct me. This is wrong. That is wrong. So having a better, good data set is the what to say. Uh, it's a rule of thumb for your model to be more reliable. So if you have lesser than 20 compounds, then if it is a local set, please go ahead. Otherwise, don't do QSR. Try to do topology based, that is molecular field based QSR, or COMSIA or COMFA, that is a conformation based or pharmacophore based models. That is kind of okay, but again, you have to clearly have a better understanding there. Sorry, the other question, let me take the other question. What if I have two different data set with different assay methodology, and can I still use the data set for no? So as Ambli already clearly mentioned, if you want to combine from different data set, ensure you have same assays for the same target and similar units. Otherwise, when you're merging, there is no meaning on combining those data. It might be for tyrosinase, but if the assays are different, of course there will be difference in the what to say uh, data. I mean control, or whatever the range or trend that is made within the data set will be different. What is different between ChemDraw and Marvin? ChemDraw is more what to say, it's similar software. I don't say much difference. ChemDraw being earlier being used as a mainly for reactions drawing, and also preparing publication valid formats. Nowadays, we don't have anything like that. Earlier days, ACS, RSC, Willy, Elsevier, they all came up with the ChemDraw template. So if you want to put a synthesis protocol or a scheme, you have to draw it in ChemDraw. No other choice. There's no other softwares available. Later, Marvin came. Marvin also supports to some extent the templates, but it has many other features more for Cheminformatics and all. So it does, for us, both does the same job. So you can use ChemDraw also. Since it was a paid software, we didn't use ChemDraw. You're free to use ChemDraw also, or Marvin. There are many other softwares available for drawing structures. So the next question is, I didn't get how data come to Excel sheet. Okay, so when she has saved it as a CSV file, or maybe a prop file, or a desk mat file. So when you open it into Excel sheet, it will first ask you what kind of delimiter to be used, because it is a normal text file. So the delimiter is normally for the MOPAC generated descriptor files. Their delimiter is tab, not comma, tab, right? When you come to PaDEL, it is generating a CSV file, so it will be comma separated. So that there the delimiter will be comma. So when you define that delimiter, that is either comma or tab, Excel automatically reads the text file and converts and put it into the Excel sheet. So that's how you can make your data visible in Excel sheet. Making it in Excel sheet helps you a lot, because you can remove, make modifications. Then as she was preparing the prop file, PRP file, where you have the names of the structures and the activity data. Now, let me tell you why she made all the names of the structures to S8001. If you are using any software, not mandatory to, but it is always helpful for us to have a standardized format. I prefer to do that. Otherwise, we will have mol1, mol2. Mol1, mol2 might be for any other data set also. So it is better that for better identification, you can give some specific unique names, and then you have an Excel sheet which refers to the original name. So this is just what to say, housekeeping work to make our data much more clean, and indexing it and identifiable later when you look at it. How to write effective SARs? Could you please specify your question? Write means, I didn't understand whether you want to design a new molecule, or whether you want to write a report or a manuscript on SARs. If you can read the paper that we shared in the link, I think Ambli has very nicely narrated on explaining the SARs based on QSAR and docking. How could we use any other could we use any other software? Oh yeah, you're free to use any other software for 3D optimization. So here we have used the example mopac. Let's say you did a molecular docking and you're bringing the 3D structures, they are fine. Let's say you don't have anything of them, you just have the 2D, just use the PaDEL, so that it will automatically convert 2D to 3D. There is an option to convert it to 3D, or use open babel or o minimize, so it will also generate the 3D structures. The question here is what geometry you wanted to use. Simply don't use some geometry. That is the one of the biggest bias when you're trying to use 3D geometry or 3D QSAR. You need to know what is the geometry. You either use the docked complex or use the X-ray crystal structure 3D geometry. What any other 3D geometry that you're bringing in, if it is not close to the real 3D structure or crystal structure, your QSA doesn't have any much meaning. It might be very close to the reality, but that's where it is. So if you wanted to design new molecules based on a QSR, ensure that your 3D optimization is much close to the perfect. Otherwise, your reliability of the newly designed molecules is not that great. So that is the reason why. So whatever software you can use, but make sure that it is very close to crystal structures, either biological complex or as it is. How to use doc poses for descriptor generation. Yeah, so as Ambli was using mol files to load it to PaDEL, the folder, the same way you do it. So you have SDF file which is exported from your docked poses. Use that SDF file in PaDEL, and then ensure that you enable the option called retain 3D geometry, and do not generate 3D confirmations, or do not do perform minimization. If you, these two options are clicked, and if you load the docked poses, so it will use the docked poses confirmation to generate the descriptors. So that is easy. Whatever she showed, the same step. Okay, so this is an answer I myself put in there. So kindly send a message to this particular phone number, 9959 81141. And when you send me a message, I'll add to the group so that you'll get to know updates about the session. If you have any technical questions, I am free to answer there. So kindly send a message, then I can add you to wherever possible. We have two groups available in two different platforms. We can add it. We can see which number on your number which is available, and we'll add you. What was the basics of selection of these compounds that you have took for making a QSAR model? I think Emily already mentioned that same bioassay, similar activity, then units, and the class of compounds. So these are the thing. And yeah, how did you choose 3712 molecule? What can you please elaborate? What is a 3712 molecule? I didn't understand. Is it a 3712 descriptors? Ambli, can you put the message if you have any audio issues? I didn't understand what they are referring to. Yeah, just let me understand. So Ambli is just. Oh, it is a model identifier. Okay, so okay, in QSARINS, as I told you, we will be generating several models based on genetic algorithm, right? Where she was talking about the mutations and generations. So 3712th model is what she selected. That is a model identifier number. That's all. So that's what I said. We have to do several iterations. It's not the first iteration. We took the 3712th iteration is what is being taken. So it is not the molecule. It is the model in the QSARINS. So QSARINS generates several models based on how much maximum number of descriptors the software can use to generate the model. So based on that, that is a 3712 number. The molecule, of course, when you go inside the table, you will be able to see each one of the molecule, how it is being bifurcated as a prediction set or a training set. Now, how to divide them as a training set or prediction set, either based on clustering or Y randomization, or even randomly, whatever you feel that you wanted to do. But there should be a reason why you're doing it, right? As Ambli clearly mentioned, each of the outlier, of course, when you're removing outliers, the model might improve, but you need to have a scientific reason behind why you are removing this outlier. Those things are very important. Had mentioned that you had used molecular docking. Could you please explain the significance of such step? I think already I have told you why we are using molecular docking, the poses for calculation of descriptors in PaDEL, because we want to use the closest biological complex crystal structure. It's not a crystal form anyway. After docking, it is a biological complex conformer. That's why. What is the benefit of constructing descriptors using Codesa Pro rather than using online descriptor generator? So as I told you, Codesa Pro, why we used it only because I worked for that group. One, second, there it uses Mopac. I don't wanted to show a command line based Mopac online, because if you are a new be coming to QSR, I don't want you to be little bit concerned, or should I know command line also? No. So you can avoid using Codesa Pro, no problem. Only then is you have a PaDEL there. Now, why I used Codesa Pro? That is a question here. So I wanted to have a quantum chemical based descriptor. For example, LUMO energy. I wanted to have a better parameterization when I'm going to calculate that LUMO energy or HOMO energy, whatever it is. They are all quantum chemical descriptors or thermodynamic descriptors. PaDEL uses MMF or MM2 based, they are force field based optimization. They are not accurate. So when you look at the accuracy level, MMF, MMX, MM2, M1, they are all less accurate. The next highest accuracy goes to semi-empirical parameterization like AM1, PM6, PM7, or RM1 also. After that, it goes to DFT, then CCSD, MP, and all those parameterization goes up, including Hattrifock after semi-empirical. So in order to use semi-empirical optimization, which is at least not so accurate, but at least closer to DFT, so that's the reason why being Mopac was embedded within Codesa Pro, we used it. Now, you might not be having access to Codesa Pro. As I told you, it's a paid version. So you can simply use Mopac. Mopac is exactly free version where it uses semi-empirical parameterization. You can use AM1 or PM6 or PM7 for optimizing your geometry and calculating the descriptors like HOMO, LUMO, energy, and others. So I hope I answered your question. Now, in order to generate those values, there's a particular keyword that to be used in Mopac. I will post that in the YouTube description, so that you need to just add that keyword to every mold file, and there's a script for that. It just runs. All the scripts, everything will be in my GitHub. Give me 2-3 days to upload all those details so that you will have. And all the data what Ambli has mentioned today, as it is mentioned in the manuscript, it will be in my GitHub. All the raw data will be available if you want to check the reproducibility. Could you explain on the 2D and 3D descriptors, stressing their effectiveness on the accurate QSAR model? As what I was telling you so far, 2D, there's no bias because 2D is 2D. Any software I use for the calculation descriptor is exactly the same. But 3D descriptor, let's say I used PaDEL, let's say I used Mopac, let's say I used X or Y software for optimizing my geometry. As my geometry changes, all my descriptors are going to change. So 3D, bit different. So in order to have a consistency there, either use 3D descriptors from docked complexes poses, so that you are taken from a data which is having a target binding studies have been done, that geometry is being used. So there is some continuity from the biological complex. Or if you have access to CCSD, that is Cambridge Structural Database, where you have X-ray structures of these molecules, if it is there. For all of them might not be there. For the ligand molecule, then take that crystal structure, don't do any optimization, just directly go for descriptor calculation. so that you're using a crystal structure. So 3D descriptors, you have to take care very carefully, otherwise there will be a lot of confusions on reproducibility. Why not use PaDEL itself to construct the 3D structure? If we are using 2D descriptors only, what is the significance of using? I think I already explained all my previous 3 answers. You have to take it. One, I had already told you PaDEL uses MMF force field based optimization, so their accuracy is very less. Therefore, if you want to have a better 3D structure close to crystal structure, close, not ever very close, close to crystal structure, then you have to use MOPAC at least for semi-empirical parameterization based optimization. Otherwise, go for docked, which when you have target. But if you don't have a target structure, of course, you cannot go for docked complexes. You have to go for optimized 3D geometry. I never recommend MMF for QSA because my purpose of doing QSAR is I want to design new molecules based on my models. If I want to design new molecules, I don't want to have any false positives or any non-reliable data in my models. So I want to have a good structure if I'm doing any 3D descriptors. Otherwise, go ahead with 2D. But if you want to learn things, of course, PaDEL is fine, no problem. But as I told you, you have to be very careful. And you have seen that when Ambli was doing this thing, all the 3D descriptors she considered from the MOPAC, which is from QSAR Pro. In PaDEL, she did not calculate any 3D descriptors. She calculated only 1D and 2D descriptors. So hope you realized that. What is the point of merging descriptors from the software? Why use both? Can you elaborate? I think I already answered this question before this question itself, initial question, Q&A session, that it is not necessary you need to merge. Why I merged? It's only because we want to consider 3D only from MOPAC and 1D and 2D only from PaDEL, because of personal choices of descriptors. And I told you the 3D descriptors from an MMF force fields was not okay with me. It might be okay with many community, but not okay with me because I'm going for designing new set of molecules. Which if to see the list of new molecules designed, please go to the paper which I have shared in the link. You will get the designed molecules. Which method is good for QSR? Very good question. It depends upon what kind of data you have. It depends upon what kind of diversity of your chemical class compounds you have. Also coverage you have. As I told you, MLR is good, but for local set of models, not for diverse set. If you are doing it for diverse set, take care of all possible statistical parameters in order to avoid the overfitting. Otherwise, PLS is something okay. PCA is also kind of okay if you want to bring in multiple descriptors together as factors to bring up the models. And machine learning models are also good. But coming to machine learning model, we cannot create an equation as what Ambli showed you today. But we can use the same software or the algorithms to write back or create a model to predict the activity for the new molecule. That is possible. But MLR is always best to teach and to learn the concept of QSA or why we are doing QSA. Can you explain, tell a short example, the use of QSA, docking for interaction dynamics for molecular simulation? Okay. I think I already told you, QSAR, the ultimate goal is one, to identify experimental errors. Very easy. QSAR will help you to identify experimental errors. Second, if you wanted to design a new molecule and predict its activity, QSAR models are available. So for example, online servers like PASS server, I hope everybody knows about the PASS server. It also same, it has a QSAR models in the backend, and they try to predict the activity for the new compounds that you're giving, right? So that's where the ultimate goal of QSAR. The third one is match pair series and making modifications on the derivatives. What could be the implications, whether it's going to enrich the activity or not? All those things QSAR is going to help you. In silico, when the target structure is not known, which design strategy is adopted? When target structure is not known, it is not only QSAR, because for QSAR you need to have activity data, right? Any activity data. It need not be biological, it could be some other physiochemical property also, but it should be some experimental data for you to have a better modeling capacity. The next method is a pharmacophore modeling. It can be either molecular field based modeling, that is 2D, and 3D pharmacophore based modeling that can be done, but you need to have a reference molecule. Always reference molecule should be X-ray structure or a co-crystal structure, mandatory. Otherwise, what is the point of doing that pharmacophore screening? The third one is the old style fingerprint or similarity based search studies. But similarity based also, please be very careful. There are several types of similarity, that is substructure similarity, superstructure similarity, identical, duplicate. There are different types, and including different indexes like Tanimoto, Euclidean indexes are there. So you should be very careful with similarity study. It might lead you to some false positives, depends upon the complex of your structures. So these are the different other methods. The lowest energy confirmation is always a bioactive confirmation of a drug, true or false. The question should be reframed with the protein or without the protein. That is also a question, right? So not necessarily always it is true. The reason is, recently I did analysis. I picked up a co-crystal structure where there is a ligand there. I did docking. Okay, I calculated that energy for that particular compound, and then within the pocket, keeping the constraints of the pocket itself, for the same co-crystal structure, I generated conformations within the pocket itself, not without the pocket, with the pocket. And then it generated better conformations with lowest energy, right? But that lowest energy did not give me a good binding affinity or good binding energy, right? So not necessarily all lowest energy conformations are always bioactive conformation. The lowest energy conformation might not be a bioactive conformation, but itself is a best conformation. When you added the word bioactive, when it goes to biological complex, that might not be the best pose or conformation. That's what, from just 2 days back when I did that experiment, that's what I understood. Molecular mechanics simulations are analogous to sorry, I didn't understand that. Molecular mechanics simulations again. For me, molecular mechanics is again MMF, all those things. So can you please give a bit more clarity to this question? Sorry. How to check the similarity of structures if you take structures from different literatures? Okay, good question. You can either use NIME. So in my NIME session, in my YouTube channel, there is a second NIME session where it is all the workflows are there in the description. It's already posted in the YouTube, so you can use NIME.